So I'm going to motivate this particular use case by imagining that I work on sleep in Drosophila. I've come across this interesting paper from the group of Mark Wu. They report that morphologically distinct categories of dopamine neurons in a brain area called the fan-shaped body promote wakefulness in Drosophila. So let's find out more about these neurons. As is increasingly the case, Wu's group referred to neurons in the Flycircuit database to define the different categories of individual neurons that they've been studying. So this is helpfully available in table S2 of their supplementary information. Here's table S2. Let's look, for example, at these medial fan-shaped body neurons. You can see here that they've noted three different Flycircuit identifiers that define this category of neuron. So we can go to NBLAST online here, and we can try and search for these particular identifiers and then use that to search a whole database. So we're going to do a one against all query. We're going to use the identifier that we were just looking at, which is this, and then we're going to start a search against that neuron. Let's click here. Okay, so here are the results of the search. They're just taking a moment to download the structures of the neurons from our server. Okay, now we can immediately look at these in 3D. Hopefully you'll see that they are indeed structurally extremely closely related to the query neuron, which is shown here in black. So we can find out more about these neurons. For example, we can view the raw data, which is essentially what we receive from Flycircuit here, and they provide other information, for example, indeed confirming that they think these are likely to be dopaminergic. You can also look at a cluster of related neurons here. So this is pre-computed by us and includes all of the structurally most related neurons in the 16,000 Flycircuit data set. So again, when you look at this, you should be able to see that there's a group here of at least a dozen neurons, and they are all very similar except for one outlier here, which seems to have some extra processes. If we go further down here, you'll see that we can look at other similar clusters. So let's look at the next most similar cluster to this one, and you'll see that these also target the fan-shaped body and the same region of the superior medial protocerebrum. So what I've shown is that you can use NBLAST as an entry point to find structurally similar neurons to neurons that have been mentioned in the literature.